Hello everyone. Today's discussion is about the magnetotactic bacteria. When we say magnetotactic bacteria, so tactis, magnet, and bacteria, these are the three words, right? So magnetic action, taxis is to navigate. So bacteria which are going to make their movements in response to magnetic field in the environment, specifically the one which is due to Earth's magnetic field. They are going to move in the direction according to that. And of course, it is going to be responsible for the motility and reaching to the particular niche or movement within the particular niche. Right. So we have to talk about these magnetotactic bacteria today. So when we have to talk about iron minerals, uh, as we all know, the formation of iron minerals happens due to biochemical processes, and it is known for several decades. So it's nothing new in that. And of course, if it is going to happen due to biochemical process, some organisms are involved which are going to synthesize magnetic ion compounds such as magnetite or ferrihydrite, so or goethite. So you have these magnetic molecules getting magnetic molecules, or rather I should say magnetic compounds of iron and other metals getting formed. So when we talk about these compounds which are formed by biochemical processes, there are many organisms, not necessarily bacteria, protozoa, but you have mollusks, you have some arthropods, you have some chordate molds, you have certain fungi, plants, and even animals also are known, which are which are involved in synthesis of various magnetic ion compounds. So when it comes to bacteria, they are able to form these magnetosomes or magnetic compounds intracellularly as well as extracellularly. And these intra and extracellular grains, you can say grains, matlab structure like a grain molecules of iron hydroxides or silicates, phosphates, sulfates, and sulfides. A wide range of these particles are seen, and nano size to huge size materials are available. When we talk about these, they have lot many applications, because the nano-sized particles, they are formed of noble metals and semiconductor compounds. So diverse applications, they have the potential applications, say uh, uh, in various fields. You have which is attracted by bacterial magnetotactic complexes, or the process understanding is one part which is of our interest, but the application of these bacteria and their products in various industries also are huge. So you have to know about these bacteria and how really they are going to work. As you are seeing in the first slide, this is the slide which shows the work done by scientists at CSR NIO Goa, right? So, as I said, ki nano-sized particles, nano-sized grain molecules of magnets or magnetotactic compounds are produced. But on the contrary to that, here you are seeing the different shapes, right? And
मम्मी
that screen the serene
राइटिंग पढ़ देन लैपटॉप चार्जिंग ले लाओगे
marine
जरा
environment, and which is almost 3 meters long in the Bay of Bengal. And they had characterized the different shapes and sizes of these molecules, and the properties also were studied. It is really interesting to see the diverse shapes, right? So here, let's let's go to the next slide. Excuse me for a second, it's not moving. Okay, anyway, so this is to talk about these bacteria. So when we say magnetotactic bacteria, they are widespread and they're motile, very motility. When I say 100 micrometer per second is the rate of these bacteria, and they are working under the magnetic field. So they have unique organelles which are referred to as magnetosomes, and they are going to harbor small nano-sized crystals of magnetic ion minerals, right? And as it is said organelle, surely it has to have a lipid membrane, bilayer membrane. So it is the envelope within which these nano-sized crystals are uh, uh, present. So when we talk about the aquatic prokaryotes, the motility is directed by Earth's geomagnetic field, and of course, it can be influenced by externally applied fields. So, for example, if I want to see the movement of these magnetotactic bacteria, can I see it in the laboratory? Yes, simple light microscope and hanging drop technique can help us to do that. So, what you use for motility is the hanging drop technique. Similar drop containing the suspension of these bacteria you have spotted on the cover sleeve. The cover sleeve is pasted on the hanging drop slide, that is cavity slide, and you're observing it under microscope. The only additional thing is you are going to hold a magnet and the parallel to the axis of the slide. So the north pole, if you hold towards the drop, very close to the drop, then you will see bacteria are moving to the edge of the drop, and which is in the vicinity of or close to the magnet. And if you rotate that magnet by 180 degrees Celsius, that is from this end to the other side of the slide, and the another pole is there, surely bacteria will start moving again back from the edge towards the opposite direction. So just such a simple experiment can make you check for the presence of magnetotactic bacteria if they are present. Of course, as other bacteria, these are ubiquitous. They are found mainly in aquatic environments in the sediments, and most of the studies reported are from various sea waters. The magnetosomes, when we talk about, they are formed of magnetite, that is Fe3O4, or gigaite, that is Fe3S4, that is sulfur compound, right? And it is enveloped in a double membrane layer. Now, these are the photographs we are seeing, that is, people are attempting and the scientists are attempting to sample these bacteria from the natural environments. There are different techniques which are used for sampling. So, of course, the size of these magnetotactic compounds, and the size of these particles, it is going to range between 35 to 120 nanometers, and it has got a very high magnetic moment per unit volume. So they are usually arranged in chains within the cell, and yes, magnetic dipole movement is very well demonstrated. As I told you, in the hanging drop technique method, you can see the alignment of bacterial cells. They are approaching the particular pole of the magnet, and you can see the direction of the bacterial cells getting changed towards the magnetic pole. When you're holding a magnet on your slide and trying to reorient the bacteria, yes, they respond to that. So when we talk about the characteristics of these bacteria, so generally they are found to be they are said to be found in the OAI region, that is oxygenic unoxygenic interface. OAI, that is what is they are called. But yes, some of them are strictly unoxygenic, but few axenic strains also are reported. The temperature, when you talk about mesophilic temperature, they prefer and pH near neutral, but it is not limited to that. Extremophiles or moderate thermophiles, magnetotactic bacteria are known, which are obtained from the hot springs in the um, which are present even in the Himalayan region or the northern Nevada region. So these are the reports by different scientists. So they have scanned the particular aquatic habitats, say Mono Lake from the California region, obligate alkalophilic bacteria which are producing these magnetosomes, they are obtained, or thermophilic, which are going to, of course, moderately thermophilic. 63 degrees Celsius is the optimum temperature for their growth, and pH near 9 for alkaline loving bacteria. So, uh, you have a diverse range of these bacteria which are known to produce these magnetosomes. Here is one example, Candidatus magnetobacterium bavaricum. It is found to constitute about 30% of the microbial biovolume. Out of whatever bacteria are present in that lake, 30% is accumulated by Candidatus magnetobacterium baviracum. So see the number, the dominance, the diversity of these bacteria. So this is from the Bavaria sediments, Lake Chimezi surface sediments in the Germany. So Novadia, then California, Germany, these are the different reports where people have been talking about these bacteria and trying to isolate them and grow them further for industrial applications. So when I say this, which bacteria are generally uh, known as magnetotactic bacteria? Of course, very first important feature is these are fastidious group of prokaryotes. They require special requirements, special culture conditions are necessary for their isolation and cultivation. So most commonly, they are associated with alpha, gamma, and delta proteobacteria classes of Proteobacteria phylum, and of course, Nitrospirae phylum. These are the two phyla which will give you diversity of these MTBs, that is magnetotactic bacteria, proteobacteria, and nitrospirae. These are the two phyla which are being explored very much. So when we talk about this, uh, these species which are we are talking about, say, either they are microaerophilic, anaerobic, or maybe they are tolerant to low oxygen conditions. So most culture species of the alpha and gamma proteobacteria classes are microaerophilic. And another important factor, that is they grow chemolithotropically. And of course, they require reduced sulfur compounds as their electron source, and chemoorganotrophically, which use various organic acids and carbon sources for their growth. So these are the conditions why I'm mentioning it, because they are going to be crucial when you design a medium for enrichment of these bacteria or enrichment of the magnetosomes in the bacteria. You should provide the specific requirements. As I said earlier, these are very fastidious group of organisms, so requirements are very specific. So when it comes to Delta Proteobacter are sulfate reducing anaerobes, and they grow chemoorganoheterotropically. So you have different examples I'm trying to give in each slide. Yes, magnetotactic bacteria have great potential for iron, nitrogen, sulfur, and carbon cycling in the natural environments. So when you go for sampling, what are the methods for sampling? Yes, they can be obtained from the sediment layers or the water at particular depth. What is the important thing is OAI, that is oxygenic unoxygenic interface. So some of them will be in the unoxygenic interface side, or some of them will be towards microaerophilic conditions but towards the oxygenic side, right? So at certain centimeter layers of the sediments, you can extract the sample with the help of bottom samplers. So here, as you are seeing in the picture, the free divers who has gone to the Bethany zone of the seawater is collecting the sample, right? So this is one method. So you can just take the bottles which are filled with 20 to 30% of the volume with the sediment, and the remainder is filled with the water. Air bubbles are to be avoided, right? And then it is observed that generally the top 5% centimeter region of the sediment gives you maximum number of these particular MATs, that is, sorry, MTBs, magnetotactic bacteria. Now, when we talk about OAI region in the water column, say pond, salt pond, or hot water pond, the sampling at that particular depth is done using the boat, right? So, depth profilers and oxygen probes, they are required. Peristatic pump is attached for simultaneous accurate water sampling and oxygen profiling. So, there are different methods how you can get the sample so that chances of getting these magnetotactic bacteria from that sample are more. Now, here are some of the organisms which are reported. Magnetospirillum, right? Then Bacillus species, Spingo aurantica species, Disulfobacterium vaculatum species. Yes, some of these species are from India as well, as you are seeing here. Burkholderia, then Microvirga, these are some of them which are isolated from Western Ghat forest soils as well. It is not necessary to have an aquatic environment, but yes, forest soils also have given some magnetotactic bacteria. And Lonar Lake, which is known as the lake formed by the crater, the in the Buldhana district, so that Lonar Lake also is found to harbor some magnetotactic bacteria. So you can observe them under light microscope as common bacterial cells, but you cannot see the magnetosomes, that is the organelle which harbors these magnetic structures, right? So if you want to see these magnetic structures inside the cell, then you have to go with the electron microscopy. So you are going to deposit your sample onto the microscope grids, right, which are going to be then washed and dried in the air, and then you are going to go for selected area electron diffraction, because particles cannot be directly seen. So you have to go with dispersive X-ray analysis in combination with electron microscopy to visualize these magnetosomes. Of course, the beauty of electron microscope is that you can observe the magnetosome membranes as well. All right. A second way is just grow these bacteria, let the magnetosomes develop into them. Then you extract those magnetosomes by breaking the cells, and then as magnetic particles, you can separate them, centrifuge them, separate them, and observe under X-ray or XRD analysis. You can do, or you can observe them under transmission electron microscopes also. Now, this is how they would look like. Now, are you able to see the specific shapes here? The C component of the diagram is showing you. See the shape of these dark color particles, right? In this diagram part B, you are seeing this oblong structure. Yeah, it is also a magnetosome, and the outer line which is covering the entire margin of the structure is seen. So this is magnetosome, and what you are seeing here is the bilayer lipid bilayer membrane which encloses that magnetic content, right? So here you are going to uh, get an idea how they can be visualized and how would they appear under transmission electron microscope. This diagram shows that how is the ancestry of these bacteria, how they originate, how do they diverge from the common ancestor. So common ancestry is phylogenetic groups not known to contain magnetotactic bacteria. Then phylogenetic groups containing magnetotactic bacteria, they are classified into two main things: proteobacteria and nitrospirae. Again, they are further classified as alpha, beta, gamma, zeta, delta, eta, and here OP3. So this is a schematic representation to show you the uh, classification of these bacteria, how they originate from the initial non MTB types. Right. Here, the bullet shaped magnetic crystals are seen, and the surrounding membrane also is very much clear. Now, when we talk about their presence, surely the diversity of biomineralization, fossilization pathways of microorganisms in the extreme conditions are very important. So, may it be hydrothermal springs, hot springs, hypersaline environments, seafloor basalts. So, these are the things where you will find the microbial activity, and magnetic bacteria are common in those. So, when I say about uh, these particles which are present in the cell, 10 to 30 particles per cell, which are about 50 to 100 nanometer long along the axis. They will be the generally arranged. So morphologically, if you talk about magnetotactic bacteria, they are going to include all different shapes. It could be rod, it could be coca, it could be spirillum or vibrio, and they may harbor these magnetosomes. So, general classification when you talk about, or the phyla which are considered as MTB members, they are Proteobacteria, Nitrospirota, Omnitophyta, I'm sorry, and then you have Desulfobacteriota, Fibriobacteriota, so on and so forth. So these are the few of them. Two or three examples are also enough if you can remember those. Now, this is what a schematic representation to which different phyla they belong to. So here, alpha proteobacteria, eta proteobacteria, zeta proteobacteria. You must be remembering this particular diagram: alpha, beta, zeta, delta, and eta. So all these are the types of proteobacteria. So that is what is beautifully shown in this particular diagram. So when I say this, the common factor is Fe3O4 magnetic particles they are produced, and you see the variation in the shape of the cells, which is symbolically shown here, right? So these are yeast-like forms. Here these are spirilla, here these are cocci, and here are the so diverse shapes you can find, right? So here are different bacteria which belong to the category that is magnetotactic bacteria, and these are the different shapes and the phyla which belong to it. When it comes to the shape of the crystal, these are the different shapes that are possible, and you can observe the beautiful symmetry, right? So the dimensions for each angle, the length, the surface, all these details are given in these diagrams. It is just to make you understand how beautiful the things are in the nature. So when it comes to isolation and growth of these bacteria, as I told you earlier, these are fastidious bacteria. They require specific nutrients, and they grow well at OAI, that is oxygenic and oxygenic interface. So what do they really require? They may be autotroph or lithotrophs. That is what we have seen. So if they are organotype, then they will be choosing organic acids, etc., as a source of energy. Otherwise, they'll be dependent on compounds of sulfur, for example, for the energy. So when you attempt to grow them in the laboratory, this is one of the reference that talks about isolation of magnetotactic bacteria, that is magnetospirillum species. What is the medium? It is sodium lactate medium. So organoautotroph, right? That is what the organism must be. It is able to utilize sodium lactate. Then ammonium chloride is there, yeast extract, K2HPO4, MgSO4, sodium thioglycolate. What is the sodium thioglycolate would do? It is required for developing the unoxygenic conditions. So it is going to scavenge the oxygen, and media will remain devoid of oxygen, which is required for the growth of these bacteria. So here ferric citrate, right, and the trace element mixture which is required for the growth of these bacteria. So here you are seeing the flask cultures or the bottle cultures, the flask cultures you can say, and then it is at the scale up semi-continuous culture in the industrial scenario. Such a big fermenters you are going to run for growth of these bacteria. What is going to differ is here people media used is feeding media and growth media. So initial growth media is very much like your laboratory media, but when it comes to feeding media, it is going to contain lactic acid, then it is going to contain ammonium, uh, NH3H2O. It is going to contain yeast extract, FeCl3, and some trace element mixtures. So growth conditions for this particular bacteria, Magnetospirillum, temperature is 30 degree, starting pH 7, airflow and stirring rate initially set at 0.5 liters per minute. So these are the details. Of course, scale up is not expected, but you need to know at least how you can enrich these bacteria, right? So here are certain details which talk about uh, particular features to be taken into consideration when you want to enrich those organisms and grow them. So generally, as I said earlier, they may be obligate or facultative chemolithoautotrophs, right? So which are chemoorganoautotrophs? They can oxidize format microaerobically, electron donor, and fixing the product CO2 by using Kelvin Benson's Vassam cycle, CVP cycle, the mode of nutrition and metabolism we are talking about. Yes, semi-solid oxygen concentration gradient medium also can be utilized for their growth. So oxygen gradient, so concentration of thioglycolate you may adjust, and semi-solid media is going to be utilized. So semi-solid media can be used in uh, shake tubes, yeah, like shake flask. Slant tubes are there, but they are shaking tube, shake slant tubes. That is what is they are referred to as. They can be used. So when you are talking about semi-solid oxygen concentration gradient media, bicarbonate is included in the media. organic compounds should be omitted in that case. So exception is that cysteine requirement and vitamin requirements. So it is not that only organic component like lactic acid you give when the requirements are done. No, certain selective requirements may be there, like trace element composition. You might have to add specific nutrient requirements like cysteine or vitamins, or maybe yeast extract can compensate for all of those. So when we talk about chemoorgano heterotrophic growth, now there are various choices. For example, succinate, acetates, some amino acids uh, are there, but no MTB have been shown to utilize any other type of organic compound. So, for example, carbohydrates or polysaccharides, no. So you have to provide organic acids as carbon source. So another important thing when you are talking about magnetosome synthesis within these magnetotactic bacteria, surely iron is going to be required. So you need to add iron in the medium. The type of iron source is not really critical, but the concentration is very critical. So what is important is whatever iron source that you are adding to the media should remain soluble and accessible for the bacteria. So the pH condition specifically should not hamper the solubility of iron source, and no other organic material should chillate out or complex with the iron so that accessibility of the iron is compromised. That is the only thing that is required. Secondly, the concentration of iron salt is very important. Generally 20 to 50 micromole is required. Depending upon the type strain, you have to optimize it. Just as an example, magnetospirillum, that is what we have been talking about till now. The iron concentration, as you increase, the more amount of magnetosomes will be seen, but greater than 100 micromoles cannot be tolerated by these bacteria. So, this is what you have to remember in when you are talking about enrichment of these magnetotactic bacteria, so for formation of magnetosomes. So depending upon whether the organism is chemoorgano autotroph or heterotroph, accordingly the media components are to be adjusted. Yes, can we grow them on the agar, solid agar? Yes, solid agar plates you can grow them, but generally what is preferred is activated charcoal agar. So that has been found to be very useful for magnetospirillum bacteria. And activated charcoal is going to like scavenge out and decompose toxic free oxygen radicals, right, peroxides, etc., which are going to inhibit microaerophilic bacteria. So charcoal agar was found to be useful. So activated charcoal agar, that is ACA, it is incubator under microaerobic or anaerobic conditions, or maybe 1% oxygen and nitrogen is added, or oxygen free gases they may be there, like you have CO2 incubator, you can purge different inert gases also, nitrogen also can be added. And the colonies can be seen growing onto the surface. So you may use the shake tubes or you may use the plates, depends whatever is available and suits for the culture. Now once these colonies are observed, you can go for their identification further. Now this is what is the schematic representation that shows when you are talking about growth of these magnetotactic bacteria. So depth from which they are collected. So we are collecting them from the seawater, right? The depth at which they are collected. What is the oxygen solubility at that particular depth? That is going to be very important. So that anoxygenic interface, that is no oxygen at all, and whatever oxygen can penetrate and be dissolved at particular depth in the water, that is going to vary location to location. So that depth is one of the important component. Oxygen is the solubility of oxygen at that depth, sulfide concentration at that depth, and ion concentration at that depth. These are the three crucial parameters when you're talking about growth of magnetotactic bacteria and adjustment of growth culture parameters. So magnetite producers, when you talk, they are microaerophilic or maybe anaerobic, right? And when you talk about magnetite preservation, then preservation by unoxidized magnetite form or reductive dissolution. These are the methods. We are not going to go in the details of this, but yes, you can preserve these magnetites. So magnetite crystals are there which are going to provide the magnetotaxis, that is movement under natural magnetic field or externally applied magnetic field. Bacteria will reorient themselves. Tactic movement, right? Taxis response to the stimulus. The stimulus in this case is the magnetic field. So this is a schematic representation just to make you understand the importance of sulfide, concentration of iron and that of oxygen while enriching magnetotactic bacteria and magnetosome production. Now here another point is, uh, are there having any specific set of genes which are going to make them special and are allowing them to accumulate magnetosomes? Yes, it is referred to as MAM AB operon. Right here you see in the second line MAM small AB capital. This is the operon. Right now, this operon is universally functioning for magnetosome chain formation. It contains about 10 genes which are named as MAM. It starts with ABE IKL MOPQ. Right. So these are the 10 genes which are important and they are conserved in all magnetic producing magnetotactic bacteria. Right. And also 9 of those 10 they are observed in gregite producing bacteria also, that is with sulfur, iron and sulfur magnets, right? So gene pool is almost similar in all of them, which is organized as an operon and it is named as MAM AB operon. So when we talk about the proteins encoded by these genes, very important is going to be biomineralization capacity, right? So of course, these MAM I and MAM L gene, they encode proteins that are MTP specific with no homologous uh, known in other non-magnetotactic bacteria. So these are very unique. MAMI and MAML genes which encode the proteins which are MTP specific. How it is determined? It is determined by blast search. That is, sequencing is done and it is matched with the database and the functionality metabolomics and that database. So the data obtained from blast search tells that the sequence similarities, but at the same time you have to conduct certain experiments like mutagenesis experiments, whether that particular function is going to be challenged by particular mutations, and that will help you understand whether this particular gene is commonly present or its divergent forms or its mutants are available or not. But what is seen is MMI and MML, they are only specific in MTB bacteria. The sequence patterns are unique in those organisms. Now, what do they do? Where are they involved in? So, MAMI and MAML are involved in invagination of the magnetosome membrane, right? The first aim is magnetosome formation. So these two things are very important, where these are going to be of important. So another thing which is important is tetra tricopeptide TPR, that is produced by MAMA gene. It participates in assembly of magnetosome membrane through protein-protein interaction. So each of these 10 genes, they have specific role to play. Diffusion facilitator is one of the important thing which is mediated by the proteins MAMB and MAM. Then you have certain uh, genes whose functions are yet uncertain and not revealed. So, for example, it is MMQ and LMA domains. The functions are not yet clear, but they are present. Some accessory rules these proteins may be playing in the entire process. So, at least four four domains, that is MMP, MME2 domains, and MMT domain, are involved in redox control of ion, that is Fe2 Fe3 stoichiometry control, right? So P, E and T, these are involved in redox control of Fe ions. Then there are actin like domains that of MMK, they are involved in magnetosome chain assembly, right? So positioning these magnetosomes inside the cell, putting that getting inserted across the membrane, organizing is one of the factors. So all these different domains of these different proteins produced by these 10 genes which are organized on MMAB operon are involved in magnetosome production, assembly and functionality. So this was an overview of magnetotactic bacteria. That is what I wanted to share with you. Last 2-3 minutes are remaining for this session to end. Do you have any queries? Please do ask. See, this is what all we have talked till now about. I'll be sharing this PPT with you.